Welcome to the Induced Fit Model of enzyme action, proposed by Daniel Koshland in 1958. This revolutionary model transformed our understanding of how enzymes interact with their substrates. Let us examine the fundamental concept. On the left side of your screen, you see a beige-colored enzyme with a flexible active site. Notice how the active site appears. Incomplete and does not perfectly match the substrate. The enzyme is shown in a warm, beige tone representing its dynamic and adaptable nature. On the right side, observe the same enzyme after substrate binding. The blue triangular substrate has entered the active site and something remarkable has occurred. The enzyme has changed its shape. The active site has molded itself around the substrate, creating a perfect fit. This is indicated by the label active site adapts positioned below the adapted enzyme. Pay attention to the arrows connecting these two states. A green dashed arrow curves from the initial enzyme to the final adapted enzyme, showing the direction of conformational change. A gray dashed arrow points from the initial active site toward the adapted state, emphasizing the structural transformation. The substrate is shown in vibrant blue entering from the upper right corner. The word substrate appears in the top right to guide your understanding. At the bottom right, a comprehensive explanation states that the enzyme active site is flexible and changes shape upon substrate binding. The enzyme molds itself around the substrate for optimal fit. This dynamic interaction is the cornerstone of the induced fit model and explains why enzymes are such efficient biological catalysts. Now let us explore the complete catalytic cycle of the induced fit model through 6 distinct steps shown across your screen. Step 1 begins on the far left. A blue enzyme with a textured surface displays its active site as a lighter blue region. A red spherical substrate approaches from the right, indicated by a red dotted arrow. The label reads free enzyme with active site. In step 2, the red substrate molecule moves closer to the enzyme active site. The proximity increases, but binding has not yet occurred. The label identifies this as substrate molecule approaching. Step 3 reveals the critical moment. The substrate has entered. The active site and red curved arrows surround the enzyme, showing conformational change in multiple directions. The enzyme structure visibly shifts and adapts around the substrate. The label clearly states, enzyme changing shape with conformational change written below. Step 4 shows the enzyme-substrate complex fully formed. The enzyme has completed its shape, change, and now tightly embraces the red substrate. The fit is perfect. This is labeled enzyme-substrate complex formed. In step 5, catalysis occurs. The red substrate transforms into green products shown as 2 connected spheres. A green arrow points upward, labeled product formation, indicating the chemical reaction has taken place within the active site. Step 6 on the far right shows the final stage. The green products have been released and float above the enzyme. The blue enzyme returns to its original conformation, ready for another catalytic cycle. The label reads, product release and enzyme ready again with enzyme. Regenerated noted above the products. This 6-step sequence demonstrates the dynamic nature of enzyme catalysis and the beautiful molecular dance that enables life itself. Let us examine 3 powerful real-world examples of the induced fit model in action. First, on the left is hexokinase, shown in purple and blue. This enzyme catalyzes the phosphorylation of glucose. Notice the 2 lobes of the enzyme in purple and blue colors. Black curved arrows indicate the closing motion as the enzyme undergoes conformational change. The substrate's glucose shown as a red and black molecular structure and ATP shown as yellow phosphate. Groups approach the active site. After binding, the enzyme closes around them, bringing the molecules into optimal position for the reaction. The products glucose, 6-phosphate, and ADP are released. This dramatic hinge motion exemplifies induced fit perfectly. In the center, observe DNA polymerase rendered in teal green. This remarkable enzyme synthesizes new DNA strands. The double helix DNA template, shown in purple and blue stripes, enters the enzyme active site. Black arrows indicate the conformational changes as the enzyme wraps around the DNA. A single orange nucleotide represents the incoming DNTP substrate. The enzyme undergoes significant shape change to position the DNA template and catalyze the addition of nucleotides. The products are elongated DNA and pyrophosphate. This example shows how induced fit enables precise molecular recognition. essential for genetic fidelity. On the right, ATP synthase appears in orange and brown tones embedded in a gray phospholipid membrane. This rotary motor enzyme has 3 beta subunits labeled with the Greek letter beta. Each subunit exists in a different conformational state labeled open in white, loose in black, and tight in black. Green arrows show the rotation of the central stalk. ADP shown as a red sphere and inorganic phosphate shown as a green circle with pi enter the loose state. As the enzyme rotates through conformational changes, ATP shown as a yellow sphere is synthesized and released from the tight state. This rotational catalysis demonstrates induced fit through mechanical movement. These 3 examples showcase the versatility and universal importance of the induced fit mechanism across different enzyme classes and biological functions. The image is divided into 4 vertical panels against a light blue background. Panel 1 is labeled Flexible Active Site. It shows a beige enzyme structure. A dotted line indicates the initial shape of the active site, while a solid line shows how the enzyme changes shape to accommodate the substrate. Panel 2 is labeled Dynamic Recognition. A blue triangle representing the substrate approaches the beige enzyme. Blue curved arrows indicate the movement of the enzyme as it wraps around the substrate. The enzyme on the right shows the blue substrate nestled inside the adapted active site. Panel 3 is labeled Transition State Stabilization. The beige enzyme holds a yellow star shape representing the transition state. Below this is an energy graph. The graph shows a high blue curve labeled IA uncatalyzed and a lower red curve labeled IA catalyzed, demonstrating how the enzyme lowers activation energy. Panel 4 is labeled Enhanced Specificity. A green key-shaped molecule fits perfectly into the enzyme. A red cross symbol floats nearby, indicating that incorrect substrates are rejected. At the bottom of the image, a sequence of text connected by curved arrows describes the process flow. Substrate approach, binding, conformational change, catalysis, and product release. This slide illustrates the advantages of the induced fit model. The first section on the left is titled Explains Conformational Changes. It shows a beige enzyme shifting its shape. Small curved lines near the enzyme indicate movement or flexibility. A green dotted arrow points from the open shape to the closed shape. A green check mark in a box appears at the bottom. The middle section is titled Transition State Stabilization. It features an energy graph with a vertical axis labeled Energy. A tall red peak is labeled IA without enzyme. A lower blue peak is labeled IA with enzyme. Next to the graph, a beige enzyme holds a yellow star shape. A green check mark is at the bottom. The third section on the right is titled Multiple Substrate Handling. It shows a large beige enzyme accommodating 3 different shapes, simultaneously a blue circle, a red triangle, and a purple hexagon. 2 green check marks are visible near the bottom of the enzyme. A green check mark in a box is at the very bottom right. On the far right, a text box reads, "These advantages make induced fit model more accurate for explaining enzyme catalysis." Koshland 1950. This image provides a detailed molecular view of the induced fit mechanism. In the center is a large cross-section view of a beige enzyme. Inside the active site, there is a blue triangle representing the substrate. The area around the substrate glows yellow. Red plus signs and blue dashed lines surround the substrate, representing electrostatic interactions. Green curved arrows surround the outer edge of the enzyme, indicating its flexible nature. Several labels with black lines point to specific parts of the enzyme. Top left, flexible active site points to the upper rim of the enzyme. Left side, conformational change points to the shifting structure. Bottom left, non-covalent interactions points to the lower section. Right side, transition state. Stabilization points to the right wall of the active site. Bottom right, substrate specificity points to the lower right rim. In the top right corner, there is an energy graph. The vertical axis is labeled energy, and the horizontal axis is reaction coordinate. A red curve shows the reaction path, starting at reactants, rising to a peak labeled transition state lowered by enzyme, and ending at products.